Hi, my name is Zaria, and this is my research project for microbiology. Antibiotic resistance is a major global health problem. It makes infections harder to treat and increases the risk of diseases spreading, severe illness, and even death. A key group involved in this problem is called the escape pathogens, which include bacteria. These organisms are known for their ability to escape the side effects of antibiotics. In our project, we use a safe, non-pathogenic relatives to Acinetobacter bailii and Bacillus subtilis to model these harmful bacteria in a lab setting. The purpose of the study was to isolate bacteria from soil, observe their growth, and test whether they produce antimicrobial compounds that can help fight antibiotic resistance. To start, we collected a soil sample from a grassy area near off-campus apartments. Then we performed serial dilutions and plated the bacteria to grow isolated colonies. From there, we created a master plate and selected different colonies to study further. Next, we performed several tests, including streak plating, gram staining, biochemical tests, PCR analysis, antibiotic assays. These steps helped us identify the bacteria and test for antimicrobial activity. Starting with Figure 1, we tested fermentation. The bacteria were positive for glucose fermentation but negative for lactose and sucrose. In Figure 2, the organism tested negative for urease but positive for starch hydrolysis, meaning that it can break down starch. Figure 3 shows growth on PE8 agar but not on McConney agar, which indicates the bacteria is gram-positive. This was confirmed in Figure 4, where the gram stain showed purple rod-shaped bacteria consistent with Bacillus. In Figure 5, the bacteria tested positive for motility but negative for indole and orthene. Looking at Figure 6, we observed zones of inhibition on the master plates, which suggests that the bacteria may produce antimicrobial compounds. And finally, in Figures 8 and 9, the antibiotic assay plates show varying effectiveness on antibiotics. Some zones show resistance while others show susceptibility. Based on all of our results, we concluded that our isolate is most likely a gram-positive Bacillus-like organism. Although the PCR results were not reliable due to the short CNA sequence, our biochemical and staining tests supported this identification. The presence of zones of inhibition suggests that this organism may produce antimicrobial compounds, which is important because it could help in developing new antibiotics. Moving forward, our research question is, does this bacterial isolate produce antibiotics that can inhibit escape pathogens? Our hypothesis is that if the isolate produces antimicrobial compounds, it will inhibit the growth of test bacteria. To test this, we will perform a zone of inhibition assay directly against escape pathogens and measure the effectiveness. Overall, this project helped us to better understand how environmental bacteria may contribute to solving antibiotic resistance. Identifying and testing these organisms is an important step towards discovering new treatments.